Hello everybody and welcome to my channel Omini Bio Entrance. Today we will be discussing the National Entrance Screening Test, that is NEST 2018 Session 1 question paper. I will be discussing only the biology section. So coming to the first question. The combination of numbered items that correctly completes all of the following statement is: transport through the phloem is dash while transport through the xylem is dash. Loading of sugars into phloem is dash at the source and movement of water out of the phloem at the sink is dash. Now xylem transport is unidirectional. Why? Because water column flows only upward due to two main forces, that is transpiration and root pressure. Transpiration causes negative pressure while root pressure is positive, and this will send water upwards towards the shoot regions from the roots. So 2 is unidirectional. So 2 is unidirectional. Now phloem carries dissolved sugars from the leaves or their site of storage to other parts of the plant that require nutrients. That is what phloem is doing. The nutrient rich region that supplies sugar for the rest of the plant are called the sources. So, and this in sources, sugar is moved into phloem by active transport, in which the movement of substance across cell membranes requires energy expenditure on the part of the cell. So 3 is by active transport. Then sink area are in need of nutrients. So movement of water out of the phloem at the sink is by osmosis, and 1 is bidirectional. So option 1 is correct. In the ABC model of floral organ determination in Arabidopsis, the expression of class A genes alone determines development of sepals. Expression of class A genes along with class B gene determine petal formation. Expression of class B genes along with class C genes determine the formation of stamens, and expression of class C genes alone determines carpel formation. Loss of B gene function would result in a flower with whorls in the following order from outer whorl to inner whorl. So they have given that in the ABC model of floral organ determination, how sepals, petals, stamens, carpels are determined in Arabidopsis. So if gene A is present, then sepals will be formed. If A along with B is present, then petals will be formed. If B plus C is present, then stamens will be formed, and if C alone is present, carpel will be formed. Now if B goes off, what will happen? A will form sepals, A plus B will form sepals because only A is present, B is not present. So instead of petals, sepals will be formed, and B plus C, it will be carpel, because if B goes off, C only will be present, and C codes for carpel. So C alone will give rise to carpels. So from the outer whorl to the inner whorl, it will be sepals and sepals, carpel and carpel. So option A is correct. Then next one, a cross between two plants with genotypes PPQQ and PPQQ was carried out where P and Q are dominant over P and Q respectively. Of the total progeny obtained, the proportion of offspring that would only express one of the two dominant alleles. So they have asked to calculate the proportion of offsprings that would express only one of the two dominant allele, that is either capital P or small q. So these are the genotypes they have given for crossing, and this is the Punnett square, and in this you can see that either capital P or Q is expressed only in 1, 2, 3, 4, 5, 6. So 6 divided by this total number of offsprings is 16. By 16 you will get 3 by 8. So answer is option C, 3 by 8. Then next one. Few statements about water reabsorption in kidney are given below. Decreased production of antidiuretic hormone, increased osmolality of plasma, decreased permeability of kidney tubule epithelium to water, increased reabsorption of water. The consequences of dehydration in an otherwise healthy human would be. So if you do not drink enough water, then serum osmolality increases. Why? Because the concentration of the various chemicals in the blood is increased. When the serum osmolality increases, what does the body do? It will start to release ADH, that is vasopressin. What will this do? It will keeps water from going out of the body through urine. How? It will increase the amount of water in the blood by increased reabsorption of water in the kidney tubules. Thus, ADH help in restoring serum osmolality at normal levels. So option 2, that is increased osmolality of plasma, is correct, and 4, increased reabsorption of water is correct. Then decreased production, no, and not decreased production, increased production of antidiuretic hormone. Decreased permeability of kidney tubule, no, it will increase the permeability of kidney tubule epithelium to water. So 2 and 4 are correct. Then next one, a deer with energy reserves of 150 kg spends 54 kg in respiration while 84 kg is excreted as fecus and urine. Based on this information, the efficiency of energy transfer from deer eaten by a lion will be. So from the data we are getting that about 150 kilojoules of energy reserve, the deer spends 54 in respiration and 84 in excretion. So the amount of energy saved by the deer will be 150 minus 54 plus 84, that is 12. Then efficiency of energy transfer from deer to lion will be 12 out of 150, 12 will be saved by the deer into, that is 12 divided by 150 into 100, that is 8%. Then next one, a person had therapeutic surgery which removed part of his stomach. Later, he started suffering from pernicious anemia, that is vitamin B12 deficiency. Biochemical test showed that he had vitamin B12 deficiency. The most plausible cause for the deficiency is. Now there is a special protein called as intrinsic factor that that binds vitamin B12 so that it can be absorbed by the small intestine. This intrinsic factor, this is produced or released by the parietal cells of the stomach. So when the stomach does not make enough intrinsic factor, then it cannot absorb vitamin B12. So it is the loss of parietal or oxynitic cells that is causing this B12 deficiency. So the part of the stomach that was removed might be the region around the parietal and oxynitic cells. Then number of chromatids present in the human cell in the G2 phase, prophase, post cytokinesis, that is after cell division, and G1 phase respectively are. During G2 phase of replication, the DNA has already replicated in the S phase. So after S phase only G2 phase come. So it will have double the number of chromosome, that is 46 plus 46, that will be 92. Now prophase also will have double the number of chromosomes, that is 92. Post cytokinesis, that is the cell undergoes mitosis, then it will divide into two cells. So that is post cytokinesis. After cytokinesis, the original number will be replaced. So it will be 46. And after mitosis, the G1 phase starts. So it will be 46 in the G1 phase. The property that is not common to mitochondria and chloroplast is the machinery to generate ATP, that is common to both mitochondria and chloroplast. The presence of double membrane. Both mitochondria and chloroplast have the presence of double membrane. Then presence of same set of electron acceptors. No, they have different set of electron acceptors. Then the non-Mendelian inheritance pattern. No. So option C is correct. It's the one which is not common to mitochondria and chloroplast. Then next one. The sequence of double-stranded DNA which produces a pentapeptide is given below. The correct mRNA sequence corresponding to the pentapeptide is, that will be the complementary to the 3' 5' end. So because I have already told that the 3' 5' end is the one which will act as the template for the production of mRNA, and the sequence of mRNA will be same as the sequence of the 5' 3' of the DNA strand. Why? Because this is the coding strand, and that is why, because it's, and in this and in mRNA the T will be replaced by U. So option A is correct. Then the Russian scientist G.F. Gauss carried out experiments on 3 species of paramecium by growing each species separately in identical culture conditions. Experiments 1, 2 and 3, as well as in combination of 2 species grown together, that is experiments 4 and 5. The results of the experiment are shown in the graphs. I couldn't include the options also here, so I have given the options in my next slide, but I will explain the graphs over here. Now what this scientist, that is G.F. Gauss, has done was, he has grown three species of paramecium in identical culture condition. This is the population density for Paramecium aurelia, this is Paramecium caudatum, and this is Paramecium bursaria. Then he has grown 2 different paramecium together, that is Paramecium aurelia and Paramecium caudatum, and Paramecium caudatum and Paramecium bursaria. Now what he observed was, there was the paramecium grow in the laboratory conditions, but after a certain density of the population, it became steady, that is it reached its carrying capacity, that is the rate of division has slowed down over here, the population is stabilizing. This is the carrying capacity for aurelia, for caudatum and for bursaria. Now when he grew Paramecium aurelia and caudatum together, he found that the Paramecium caudatum declined to extinction when grown with Paramecium aurelia. Because, why? Because Paramecium aurelia and Paramecium caudatum shared the same realized niche, and Paramecium aurelia outcompeted Paramecium caudatum for the food resources. That is why Paramecium caudatum population density has declined and that of Paramecium aurelia has increased. And when he grew Paramecium caudatum and Paramecium bursaria, he found that both the species are able to survive. Why? Because the niches of the 2 species do not overlap here. So they are able to avoid competition and thus able to survive. So in experiment 5, when he combined Paramecium caudata and Paramecium bursaria and allowed both of them to grow in the culture conditions, they both were able to grow. Why? Because their niches do not overlap each other. So we will come to the option. The carrying capacity of the culture media is identical for all the 3 species. No. Paramecium caudatum and Paramecium aurelia share the same realized niche. I already told that. When grown together, Paramecium aurelia utilizes resources more efficiently than Paramecium caudatum. That is why its number increased. So that is also correct. Competition has a negative effect on the growth of Paramecium bursaria and a positive effect on the growth of Paramecium caudatum. Nothing like that. They both were growing at their own niches, and both the niches do not overlap each other, and they were able to avoid competition. So option D is wrong. Then in biological system, proteins, that is repressors and activators, control gene expression. Repressors bind to promoters and inhibit gene expression, as you have seen with the studies of lac operon. Repressor will bind to the operator and it will block RNA polymerase from binding to the promoter, thereby inhibiting the transcription and translation of the structural genes. Then some promoters require activators for gene expression. These constitute a genetic circuit and are analogous to logic gates in digital electronics. A schematic diagram of genetic circuit in a cell is shown below. Binding of IPTG to LAC-I activates transcription from PLAC. Binding of arabinose to ARA C activates transcription from P ARA. So what they have, here also I couldn't put all the options here, so I have put it in the next slide. But I will explain what's going on over here. Then binding of IPTG to LAC-I activates transcription from PLAC. Now IPTG will bind to LAC-I and it will prevent binding of LAC-I to PLAC promoter, and this will result in the synthesis of the protein P. Similarly, arabinose ARA that will activate this ARA C, it will bind to the P promoter ARA, that is PARA, and this will result in the synthesis of the protein Q. Now P and Q, they both together bind to PNST and it will result in the production of green fluorescent protein or GFP. So both P and Q are required for the production of GFP. Now coming to the option. GFP is made in the presence of IPTG and arabinose. That is why P and Q are required. Only if IPTG is present, protein P will be produced, and if arabinose is present, protein Q will be produced and GFP will be made. Then GFP is made if either IPTG or arabinose is present. Nothing like that. For GFP to be made, both IPTG and arabinose has to be present. This is an example of genetic AND gate. That is also correct. This is an example of genetic NOR gate. That is wrong. So option A and C are correct. Now next one. The graph below shows the Michaelis-Menten kinetics for two enzymes reaction, that is R1 and R2, represented by as solid and dotted lines respectively. Enzyme E1 and E2 have Vmax values Vmax1 and Vmax2 respectively. The respective Km values for E1 and E2 are K1 and Km2, and Km1 is equal to Km2. The correct statement for reactions R1 and R2 is or are. E1 and E2 can never be the same enzyme. Yes, E1 and E2 can never be the same enzyme, but they can be different concentration of the same enzyme. That is why they have different value for Vmax. Because the value of Vmax is dependent on the amount of enzyme used in the reaction. Double the amount of enzyme means double the Vmax. Hence, E1 and E2 may represent two different concentration of the enzyme. That is why as the concentration of enzyme varies, the Vmax also varies. Now, E1 and E2 may be the same enzyme acting on two different substrates with different affinities. Now, the Km can vary greatly for different substrates of the same enzyme. But they have given that Km1 is equal to Km2, which shows that it is the same substrate. Then, the E1 and E2 may represent two different concentration of the same enzyme acting on the same substrate. So that is correct. E1 and E2 may be two different enzymes with the same affinity for the substrate. Yes, because the Km can vary from one enzyme to another. But they have given that Km1 is equal to Km2, which also means that the same affinity for the substrate even though the enzymes are different. So Km1 value will be equal to Km2. So option D is also correct. Khorana performed experiments to decipher the codons that code for a particular amino acid. The results of these experiments using synthetic mRNAs and the corresponding polypeptide repeats that were generated are given in the table below. Based on the results, the correct combination of codons and amino acid is or are. So they have given some synthetic RNAs, that is CAA n times, that will result in the synthesis of threonine, ASN and GLN n times. Now I have done the I have discussed the answer in the next slide. Now CAA n times, that is CAA, CAA, CAA. Now CAA, if you take, you can get codons CAA, AAC and ACA. These can code for 3 different amino acids, that is threonine, asparagine and glutamine. But we don't know which codon codes for threonine or which codon codes for asparagine or which codon codes for glutamine. Similarly, UUC enzymes you can have UUC, UCU, CUU, serine, leucine, phenylalanine. But which codon codes for serine or leucine or phenylalanine, we don't know. But we are conducting other experiments like UAC enzymes. UAC enzymes you get UAC, ACU, CUA, that codes for threonine, leucine and tyrosine. But which codon codes for what, we exactly don't know. Similarly, UUAC enzymes you will get four different codons that may be coding for leucine, threonine and tyrosine. Now if you look at all the four of these experiments that were conducted, you can see that CUU is present here as well as here. So it might be coding for leucine. So CUU for leucine may be correct. Now if you see for UAC is present here. So wherever tyrosine is present, UAC is present, and here also UAC is present. So UAC will code for tyrosine. That is why I have given a pink sketch color for that. Then UUA might code for leucine because we have got leucine, the threonine and no, threonine we didn't get, and tyrosine values. So UAA might be for leucine and ACU. So here also ACU is present. So that may be coding for tyrosine. But here the leucine is coded by CUA. And it is UAA here. So as per the option, UAA is leucine. So leucine will be coded by UAA and ACU will be coded by threonine. And UAC is already by tyrosine. So options CUU leucine, ACA tyrosine and UAC tyrosine are correct, as well as option UUA leucine, ACU threonine and UAC tyrosine are Now next one, the condition that allows a population to exist in genetic equilibrium is SORA. Mutation leading to an increased fitness should occur. That will not lead to genetic equilibrium. Then next one, migration of individuals should be prevented so that alleles found in one area may not get removed from or introduced into other areas. That is correct. All individuals in a population should have equal chance of survival. That is also correct. Then random changes in allelic combinations occurring during meiosis should get absorbed without any scope for their preferential multiplication. That is also correct. So thank you. Thank you for watching. Please like, share and subscribe. Thank you.